Welcome back, everyone. I hope you all had a nice break. Sorry about the confusion with the timings. I hope that it didn't cause anyone too much inconvenience. So we're going to start now with comparative genomics. I'm going to give a quick overview of the comparative genomics data that we have in Ensembl, and then we're going to move on to the demonstration, and I can show you where you can find various different comparative genomics resources in Ensembl. So let me just jump into my slides. So it's a very exciting time for comparative genomics, as there are a lot of large consortia that are aiming to sequence all living organisms. For example, we have the Darwin Tree of Life, which aims to sequence all eukaryotes in the British Isle. There's also Vertebrate Genomes Project, which aims to sequence all vertebrates on Earth, and Ensembl is heavily involved in both of these projects. So the objectives of comparative genomics is to examine evolutionary relationships and find differences between the genomes, but more importantly, the practical applications are to drive gene function based on homology, so we can learn something about the gene function of a less studied organism or more obscured species by looking at its equivalent gene. In better studied organisms, but also the other way around. So we can learn about the gene function in humans by manipulation in model organisms, and all of the animal research is based on this assumption of homology. So comparative genomics is used to identify highly conserved regions, which tend to be similar across taxa, as they are highly functional, and any new variation would likely be deleterious in these regions, and this is being removed by purifying selection. So we perform comparative genomics analysis by taxa. We have vertebrate comparative genomics analysis in Ensembl, which also includes a few non-vertebrate model organisms. So these basically serve as an outgroup route to the tree, and then you could find comparative genomics analysis in Ensembl genomes done separately for plants, metazoa, fungi, protists, and there is unfortunately no bacteria comparer, as there's just too many of them. You can find comparative genomics analysis on the gene tab. So I'm showing you here as an example the plant gene tab. So you can find on the menu on the left-hand side that there is a section that is dedicated to comparative genomics analysis. But then we also have wider comparative genomics analysis across taxa, which is called the pan-taxonomic comparer. And you can see it also includes a selection of some bacterial strains over here. It's not done for all species in Ensembl and Ensembl genomes, only for a subset of representative species across all divisions. So the pan-taxonomic comparer can also be found in the gene tab, but this is only available for Ensembl genomes. So if you are working with vertebrates and you would like to look at this wider pan-taxonomic comparison, you can get there by a non-vertebrate ortholog of your gene of interest. There are different comparative data types that you can find in Ensembl. So we can distinguish gene-based resources, which can be found in the gene tab, and I will show you these in a minute. We have phylogenetic trees, like, for example, protein trees and non-coding RNA trees, but also homologies that are inferred from gene trees. We also have stable ID mappings. The second type of comparative analysis data that you can find in Ensembl are called the sequence-based resources, which can be found in the location tab. And here we have analysis derived from whole genome alignments. So for example, reconstructed ancestral sequences. We have base scores such as age of base, conservation scores, constraint elements, but also syntonies, which is a conservation of gene order on a chromosome. And what I have been calling gene trees are actually protein trees. We don't calculate gene trees for all transcripts in Ensembl. What we do is we take a representative set of translations. So we use one translation per gene from all species, and the representative translation is called the ensemble canonical transcript. So we talked about ensemble canonical transcript yesterday. And then we perform all versus all model search to classify these representative translations into families or clusters. We then calculate a multi-protein alignment for those clusters, and then the alignment we calculate, and then from the alignment we calculate phylogenetic trees for each cluster, and we also perform reconciliation against NCBI taxonomy. And we also finally infer homologies from trees, so we would infer orthologs and paralogs, and here we have a similar pipeline for non-coding RNA trees. So there are different types of homology relationships. So here we have a simple tree, and all of these genes share evolutionary origin, and speciation events are shown in blue, while the duplication events will be shown in red. So you can see that we have a duplication event in primates, and as a result, we have 2 copies of these genes in chimp and in mouse, in human and chimp. And genes which emerge through speciation events are called orthologs. So there are different types of orthologies. There's one-to-one orthology. So for example, C1 in chimp over here, and H1 in human, which is one-to-one relationship, as there is 1 copy of this gene in each species. But we can also talk about one-to-many orthologs. So for example, this M in mouse has 2 homologs, so 2 orthologous genes in both human and chimp. So this M gene in mouse is homologous to both H2 in human, but also H1 in human. Finally, genes that emerge through duplication are called paralogs. So we have duplication just over here. So for example, here we can see that C1 in chimp is paralogous to C2 in chimp. You can see that they both diverge through this duplication event. And same goes for H1 in human and H2 in human, as we have 2 copies of each gene in both human and chimp. So these are examples of within species paralogs. We can also distinguish between species paralogs. So for example, C1 in chimp will be paralogous to H2 in human, as these are defined by the event that caused them to diverge rather than the species. And you can see that C1 and H2, so C1 over here and H2 over here, have also diverged through duplication. We have 2 different representations of gene trees in Ensembl. So we have the standard gene tree, and then we also have the gain-loss tree. So this is the gain-loss tree on the left-hand side, and then we have a standard tree on the right-hand side. So this is the gain-loss tree, basically shows the exact same data as the gene tree, but just a different representation of it. So this gene gain-loss tree is kind of a more collapsed version, and it shows you the number of members in a given clade, while the regular tree gives you all of the information. Okay, so we're going to now move on to the demonstration. So I'm going to look at the GH1 gene in Atlantic salmon to find gene trees and homologs for this gene. So we're going to go to, I'm just going to move the navigation bar out of the way, and we're going to go to Ensembl. So it's on ensembl.org. And again, from this drop-down menu, let's look for Atlantic salmon. So it's just over here, and then we're going to type GH1, which is our gene of interest. Let's click on Go. I'm going to click on this first search result. And now we are on the gene tab. So we have explored this tab previously. And for now, we're going to focus on comparative genomics on the left-hand side over here. So you can see under comparative genomics, we have genomic alignments, gene trees, gene gain-loss tree, orthologs, and paralogs. So let's click on gene tree to view the gene tree for this gene. And here we have our gene tree. So you'll notice that gene trees have their own ensemble stable IDs. So we have ENS, and then GT stands for the feature or the item. So this is gene tree, followed by an 11-digit unique number. And then we have some information about the gene tree on the number of genes, speciation nodes, duplication, and we have the gene tree over here. So we have these collapsed funnels for the different sort of species over here. And then on the right-hand side, we have alignments. So green would indicate alignment, whereas white would indicate gaps or no alignment. And then we have a legend for this gene tree. So you'll notice that our gene ID of interest is shown in red. So this is over here, Atlantic salmon. And then the nodes will be shown in these different colors as well. So blue is speciation nodes, red is duplication node, cyan is ambiguous node, and so on. And then also on the legend, you can find these alignments. So you can see that white is sort of less than 33% of the sequence is aligned. Then light green would mean that about 33 to 66% of the sequence is aligned, and then more than 66% of the sequence aligned would have this dark green color. You can also expand these funnels if you'd like to view basically all of the species that are contained within this. Now on the left-hand side, we're also going to look at the orthologs for this gene. So I'm going to click on orthologs on the left-hand side. Okay, so this page shows us two tables. The first is a summary table where you can select taxa of interest. It also gives you a summary of overview of ortholog types across the different taxa. And then in the second table, we have all the orthologous genes across all species in reference to our species of interest. And you can see we have the type of orthology here, ortholog, target present ID, Curie present ID. We have these gene order conservation scores, whole genome alignment coverage, high confidence, and so on. Another thing that I would like to show you is the sequence-based resources on comparative genomics that we have, and we can find these on the location tab. So I'm going to toggle to the location tab, which is next to the gene tab. And from here, we can add some comparative genomics data tracks. So if I click on configure this page on the left-hand side, and then also on the menu of this configuration, you can find comparative genomics section over here, and you can find some multiple alignments, some conservation regions data tracks, and also some BLAST-C and LAST-Z alignments as well. So these are pairwise alignments. So let's add this 65 fish EPO extended, and so these are multiple alignments, and also these conservation scores for the 65 fish EPO extended as well. Once we're happy with these data tracks, we can click on save and close. And I'm going to wait for these data tracks to appear. Nothing happens. Let's try again. I'm going to click on configure this page. Actually, let's try these Japanese Metaga HDRR, BLAST-C and LAST-C pairwise alignments, and turn this on as normal. Save and close. Okay, I do apologize. For some reason, we cannot display the track on this last image, but in theory, as I've mentioned, that there is options to add data tracks related to comparative genomics onto your image, and then you can display alignments onto your region of interest. So that was my short introduction on comparative genomics. Let's try some exercises now. So these exercises are available on Comparative Genomics tab on the training page. I will share the training page with you again on the Zoom chat. And we have two exercises over here. So one of them is in European CBAS, and then the next one is for a turbo gene. So I will give you until 2 p.m. BST to try these exercises. And then if there are any questions, please do let me know.